I'm going to go through chapter 10, part of it, which is just the kind of the meat of chapter 10. And chapter 10 is all about antimicrobial agents. So first off, we're going to cover some of the history of the antimicrobial agents, otherwise known as chemotherapeutic agents or chemotherapy. You may have heard of that in reference to cancer type drugs, but these are chemotherapeutic drugs, just mean that they are drugs that act against disease, and infectious disease is included. Antimicrobial agents obviously are drugs that will be used to treat infections, and Paul Ehrlich was the one that first came up with the idea. He thought about magic bullets, which would be chemicals that would target to kill the microbes but not hurt the host. And he tested many hundreds of different agents, and he wasn't all that successful, although he did discover arsenic compounds that were used to kill trypanosomes, and we've seen those in the lab, and another arsenic compound that worked against treponemes, which caused syphilis, which was a huge scourge during that time. But obviously we know that arsenic is a poison, so his search for magic bullet really wasn't all that successful, because the compounds that he ultimately was able to employ was harmful to the host, which was not something that was desired. We know Alexander Fleming in 1928 came back from a little vacation and found a big mold growing on his plates in the laboratory. And just like this picture here, you see that there's a big zone of inhibition right around the mold where the bacteria is not growing. And he wondered what was causing that. Instead of just tossing his plate out, oh, it's contaminated, let me just toss that. He investigated and ultimately found penicillin, which is the chemical that came from penicillium, the mold. But it was quite a while before penicillin was actually able to be purified in large enough amounts to be useful to treat infections. Meanwhile, Selman Waksman came up with the term antibiotics, which included the antimicrobial agents that are produced naturally by organisms. That's the definition we use now, right? Antibiotic is really an agent produced by one microbe to inhibit other microbes. So going along in the history, Gerhard Dominik was the one that discovered sulfanilamide, or the sulfa drugs, in 1932. And these drugs came onto the market before penicillin did. They were much easier to manufacture. So they were used to treat really the soldiers coming back from war. We know that the semi-synthetics are the drugs, we've talked about this in class, that were altered from the naturally occurring antibiotics, and they're altered in a way to make them more effective, right? They're more stable, or they resist being broken down by enzymes, things like that. The synthetics are completely synthesized in the laboratory, and the modern tools of science have allowed us to develop drugs that really we weren't able to figure out before this time, because now we can envision what a target looks like and what a inhibitor might be to sit in the target site, that sort of thing, and so we can design different chemicals and test them out to see if they work as hypothesized. The antibiotic, the natural ones produced by one organism to inhibit another, but the terminology is used a lot more loosely in public, and antibiotic encompasses many more things, right? Antibiotic and antimicrobial are used more interchangeably, but if I ask you a specific definition, I'm asking you about the real one, secreted by an organism to inhibit another one. Antibacterial drugs, drugs that target bacteria, are the most numerous, and there are fewer drugs to treat eukaryotic infections and even fewer to treat antivirals. And if you think about it, it's because eukaryotic cells are like our own, so it's harder to find a target to harm those cells and not our own. And the viruses are using our cells, so we have to find something very specific to the virus and not to our cells in order to target those infections, and it's a little more difficult to do that. So in this particular slide, this question is posed: why aren't antibiotics effective against the common cold? I hope you realize that the common cold is a virus, and viruses are not going to be treated against those antibiotics, those things that are secreted by other organisms like penicillin. So when individuals go to their physician demanding an antimicrobial because they need something to treat a cold, there really isn't an option to treat a cold. And so they're not effective, and they're actually contributing to resistance, which we'll talk about later. Okay, so mechanisms of antimicrobial action. If we are going to successfully use a drug, then the drug has to be selectively toxic, like that magic bullet, right? It's going to harm the infecting organism, but not the host. If we think about what we were just talking about, there's many more bacterial drugs that work against bacteria, fewer against the eukaryotic infections like the protists, and even fewer against viruses. Okay, so this slide is very important. Make sure that you are noting it. It's covering the 6 mechanisms of antimicrobial action. Okay, so these are the modes of action that a drug can take on a microbe to inhibit or kill it. So let's go through them. The first one is inhibition of the cell wall synthesis right here. Okay, so because many cells, like bacterial cells or fungal infections, that sort of thing, those organisms have cell walls and we do not. Drugs that inhibit the cell wall synthesis is going to cause the cell to be very sensitive to osmotic changes, and therefore they are going to die under stress. So there are a lot of drugs that fall into this category of inhibiting cell wall synthesis. Probably the second most common type of drug category are the drugs that inhibit protein synthesis. So because bacterial ribosomes are different than our cytoplasmic ribosomes, then drugs that can target the bacterial ribosomes will not harm our own cells. Or for the most part, right? Because our ribosomes are different except for in our mitochondria. So therefore, the drugs will inhibit protein synthesis in an infecting bacteria, but not in our own cells. So that is a very common category, inhibition of protein synthesis. The next category is disruption of the cytoplasmic membrane. So again, the drugs would have to target something in the cytoplasmic membrane that was different compared to our own cytoplasmic membranes in order to be effective against the bacteria and leave our cells largely unharmed. So it's targeting something in the cytoplasmic membrane. So that's the third mechanism of action. The fourth mechanism of action is inhibition of some metabolic pathway. So these microbes have metabolic pathways that differ from our own, and if we can find a drug that interferes with that pathway and prevent them from doing something that's necessary for their survival, then we can inhibit their growth and not interfere with our own cells' growth, because this metabolic pathway has to be something different than our own. The fifth mechanism of antimicrobial action is inhibition of DNA or RNA synthesis. So typically there's going to be a drug that interferes with some enzymatic process that's occurring during DNA or RNA synthesis that is obviously different enzymes than our own, and they can prevent the bacteria from being able to replicate, or pathogen of whatever sort. And then the last mechanism of action is inhibition of the pathogen's attachment or entry into the cell. So in order to initiate infection, attachment is the first step. Has to adhere to the cell or tissue in order to initiate infection. See if you could prevent that first step from happening, you're going to prevent infection. So those are the 6 mechanisms of antimicrobial action. So please pay attention to those. Keep good notes on those 6. Okay, so when we're thinking about prescribing an antimicrobial drug for an infection, there are a few characteristics that make an antimicrobial ideal. First, it's readily available. Okay, so you have to be able to get it, and just the fact that it exists but you can't get it isn't going to be helpful. We don't want it to cost too much, right? We want it to be inexpensive, and we want it to be stable. If it's not stable, that's not helpful, because it's going to break down before you can administer it. You want it to be easily administered, and we'll talk about the different modes of administration, and non-toxic and non-allergenic. So that's sort of that magic bullet idea. And then lastly, you would like to be selectively toxic against the pathogen and not harm the naturally occurring friendly microbes that are in the individual that is experiencing the infection. And that's kind of a tall order, but we'll talk about that in a minute. Antimicrobials are characterized by spectrum of action. They're termed either narrow or broad spectrum antimicrobials. Narrow spectrum means it's only going to be working against a few types of organisms, and broad means it's effective against many. And you might think that, well, we would want a broad spectrum antimicrobial, and that's usually where physicians go first. But if we're thinking long term and stewarding our choices of antimicrobials far into the future, it would be much better to be using the narrow spectrums whenever possible, because we're going to prevent the development of resistance more often that way. I'll talk about a little bit in a minute. Okay, so this is not for memorization purposes. This is just to illustrate the idea of broad and narrow spectrum. So if we're looking at something like over here, like these polymyxins that are right here, it's only going to work against, a subset of gram-negatives. That makes it a narrow spectrum. Where if you look at something like erythromycin right here, it is effective against a wide range of different types of organisms, and therefore it's broad spectrum. So ideally, you want something to be effective against the pathogen that is causing the problem and not going to affect the other organisms that are part of the normal microbiome of the individual. So a narrow spectrum antimicrobial would be preferred for that reason. Okay, so how do we figure out which antimicrobial is going to work for a particular individual's infection? Let's look at some of the tests that we do, and we've already talked a little bit about the MIC and the MBC types of analysis. Looking at this slide here, we can see the Kirby-Bauer disc diffusion, which we will be doing in the laboratory. So you can see these impregnated, they're discs, they're impregnated with antimicrobials, and it's plated on a lawn of the organism. So if the organism is somewhat inhibited by the antimicrobial, you'll see this clear zone around that antimicrobial, and if it's not, it grows up against the antimicrobial. So how do you know what is effective? Well, you actually have to measure these zones and compare it to the different charts and decide if the zone is big enough to be effective. Obviously, the bigger the zone, the more likely it is that it's going to actually be scored as an effective antimicrobial, but little zones or no zones may not be effective. And you can actually determine an effective dose with these types of analytics over here. If you look at this stick right here, it has different concentrations of the antimicrobial being tested all along this particular stick, and you see that there's a tapering off of the inhibition. So as the concentration goes down, the organism is no longer inhibited by the antimicrobial. So you can determine exactly what concentration you would need in order to inhibit the particular organism being tested. And so that would be a dose issue. Down here is an illustration of a minimal bactericidal or bacteriostatic assay, and the ones that have the turbidity, right, the cloudiness in it, are not being inhibited, and the ones that have clear wells are being inhibited. So you could determine what that minimum bacteriostatic concentration is by doing an assay like that. This is showing the minimum bactericidal types of analysis. So remember, we talked about this before. You have different concentrations of the antimicrobial in the media, and you see if there's any growth. Excuse me. So these 3 tubes show no growth, and then they're plated on media without the antimicrobial, and you see if you have any growth. So even though we had no growth at 8 micrograms per mil, shown right here, when we plate it, we actually do see growth. So that means that this is actually a minimal inhibitory concentration, whereas over here we have a bactericidal concentration, because when you have the organism growing in this tube, there was no growth, and when you plated from that particular tube, you still saw no growth. So over here at 16 micrograms per mil, this is a bactericidal concentration, and 8 micrograms per mil was a minimal inhibitory concentration. Okay, so I hope that that is clear to everybody. So I mentioned that there are routes of administration. You probably knew this without really thinking about it before, but antimicrobials can be administered in different ways, right? Delivered in different ways. So the easiest would be something that could be, put topically. So if you had a little skin infection per se, and you put something like Neosporin on it, that's topical application of the drug. Or you have an eye infection, you put a drop, eye drops in your eyes. Okay, so that's topical. Oral is when you basically swallow a pill or a liquid, and then it's absorbed from your GI tract into the blood. So oral is the simplest in order to get a systemic concentration, right, throughout the body. But you're going to achieve lower drug concentrations than in the next couple of mechanisms we'll talk about. Okay, so oral is easy. Intramuscular requires a shot, right? You're going to have to give somebody an injection. And so you get this little quick increase that you see over here, right? And then it tapers off over time. So if you give someone intramuscular, you get a little bolus, and then it tapers off. Whereas oral, it takes a while, kind of ramps up, and then it slowly comes back down, right? And then we have intravenous, and that requires, something, a needle or a catheter into the circulatory system, right, into the vein typically. And what you'll see is that when you start administration of the drug, the blood concentration is going to shoot up, and it's going to stay at that higher level until you stop administering the drug, and then it will clear from the system. Okay, so these are the different mechanisms or different methods of administering antimicrobials. When considering the safety and the side effects, there are a couple of things to keep in mind. So, drugs can be toxic, right? They have a toxicity associated with them, and that is what gives you the side effects of the drugs. Sometimes the exact cause is not really understood, and drugs could be toxic to, the kidneys, the liver, nerves, even things like tendons, right? So I'll give you a little history that might help you understand. So I have lung problems. You probably have heard me coughing. And so my lungs are kind of in bad shape. I get frequent infections. A few years ago, I had to take Levaquin, which is a antimicrobial that has a warning that says that it's very uncommon, but it can cause tendon rupture. Well, I was on Levaquin. Had been on Levaquin for a while, and I was on at home over Christmas Eve, wrapping my last present before some guests arrived, and then I went to stand up, and boom, my Achilles tendon just ruptured right there. So, caused me to have to undergo surgery and months and months of rehabbing in order to get me to walk normally again. So, that was a side effect that was associated with Levaquin. They don't even really know why it causes tendon rupture, but it does in some people. Okay, so that's a toxicity side effect. Allergies can be another problem. So they can be very rare, but they could be life-threatening. I mean, it could be something like hives, but sometimes people undergo anaphylactic shock after being administered something that they're allergic to. And then we will talk more about this in a later chapter, but antimicrobials will disrupt your normal microbiome. So it's going to kill organisms in your gut and in other places when you're taking these antimicrobials, and that leaves you vulnerable to other infections because your normal flora are organisms that are going to compete for space and nutrients, and when they're gone, then other organisms can come in and establish themselves and cause a problem. So you end up with a secondary infection. Okay, so the therapeutic index is the ratio of the dose of drug that can be tolerated compared to the drug's effective dose. And you want that to be fairly large because you want it to be effective at a lower dose and toxic at a very high dose. And not all drugs are like this. Sometimes you have things like vancomycin, which is a drug that has to be administered IV, and that therapeutic index is not very high. So what happens is if somebody's being administered vancomycin, they have to monitor the blood concentration of the patient, because if vancomycin goes too high, you risk damaging the kidneys and liver of the patient. So you have to pay attention to that. So the therapeutic index, you really want the drug to have a very low toxicity, essentially. Okay, one of the big problems with antimicrobials is the development of resistance. And so this is really just evolution happening in a small environment, like a little microenvironment. You have a patient that is taking the drug. It's going to wipe out all of the organisms that are sensitive to the drug, the ones that are naturally slightly more resistant because they have some variation because of mutation, or perhaps they have acquired a resistance plasmid or something. They're going to remain, and then they're going to be the organisms that get passed on to the next patient, perhaps. And if patients are not taking all of their course of antimicrobial drugs, they're going to increase the likelihood of survivors being passed on to the next patient. So that's a problem, right? So resistance is acquired by either new mutations or by some sort of horizontal gene transfer, right, of resistance plasmids that can happen via transformation, transduction, conjugation. Okay, so that's how resistance arises. And this little schematic on the bottom is showing you, like, you have organisms that are either sensitive or resistant. The sensitive ones will be killed off by the antimicrobial, but the resistant ones will remain. And that is what the population will look like. Only the descendants of the resistant ones will survive, and they can acquire even more mutations or other genes to help them resist other drugs and become very hard to treat over time. So that's what's been happening basically since the moment antimicrobials started being used, then the organisms started to become resistant. Okay, so at the beginning of this presentation, we talked about mechanisms of antimicrobial action. Now we're going to talk about mechanisms of resistance. So please make sure you keep those things straight. Sometimes students confuse those two, and I'll ask on an exam about one or the other, and students will just blurt out a list and it's not the right list. So make sure that you are paying attention to what's being asked, and so that you can answer. One list, if you want to say a list, it is the modes of antimicrobial action. Now we're talking about how microbes resist those antimicrobial drugs. Okay, so the first thing that we need to talk about is that they can produce an enzyme. So the example being shown below is organisms can acquire a gene that contains an enzyme that will break the antimicrobial down. So beta-lactamase is a pretty famous enzyme that organisms can acquire, and it attacks penicillins and the drugs that are like penicillin, and it will break the little ring, which is shown there in green, and inactivate the penicillin, so the penicillin doesn't work anymore. So if the organism acquires an enzyme, it can destroy the drug or deactivate it, so the antimicrobial doesn't work. So that's the first way. The second way is to slow or prevent the entry. So they can alter, maybe mutate those channels in their membrane that allows things to diffuse into the cell. And so by altering that, they can prevent the drug from getting in. They can also alter the target of the drug. So the drug might not be able to bind to the target. So if the organism has a mutation in the target may still be functioning, but it doesn't bind the antimicrobial, so it's not inhibited by it. They may alter their metabolic chemistry. So if that metabolic pathway is being inhibited in some way, they might somehow compensate for that and overcome that inhibition. They may pump the antimicrobial out. This is actually how the chemotherapeutic drugs that target cancer are often not useful anymore, that the cells mutate their pumps, and the pump will start to pump the drug out just as fast as it diffuses into the cells. Okay, the sixth example is actually very specific, but in general, this is what would happen to circumvent the inhibition of DNA synthesis. So the organism that causes tuberculosis, Mycobacterium tuberculosis, produces a protein that will bind to DNA gyrase, which happens to be an enzyme that participates in DNA replication. But when this protein binds to DNA gyrase, then the drugs like Levaquin that I just mentioned earlier, that is a fluoroquinolone, can no longer work. So by producing a particular protein which interacts with the target of a drug, it prevents the drug from working. And in this case, this is a drug that would inhibit DNA gyrase, which participates in DNA replication. And then the last mechanism of resisting antimicrobial drugs is biofilm formation. So we've talked about biofilms before. A lot of infections are caused by biofilms, and the antimicrobial drugs may kill off a lot of the organisms that are in the biofilms, but those deep down in the biofilm can be protected against the drug, and so then when you stop taking the drug, those organisms can remain. So that's what's happening even in my lungs and in lungs of patients like cystic fibrosis patients. There's organisms that are just hanging out there, and they might not be causing too much of a problem, and then something sort of happens, weakens your state, and then they're able to kind of cause an infection. But you can never really get rid of them entirely, because those organisms that are deep in that biofilm just kind of are protected, and they just are going to exist for a very long period of time. Okay, now we're going to talk about resistance to antimicrobial drugs. There's a couple of things to keep in mind. is multiple resistance, meaning that organisms won't just be resistant to one thing, but to many, or at least several antimicrobials. And sometimes when they develop resistance to one, they're resistant to others, and that's cross resistance, so related antimicrobials perhaps. And when those are plasmids, those resistance plasmids, meaning they're like fertility plasmids with genes on it for resistance to antimicrobials, when they are being exchanged from one organism to another, oftentimes they contain more than one resistance gene, and even if it's one gene, it may confer resistance to a whole category of antimicrobials, just not one. You see these multiple resistance and cross resistance appearing in hospitals and nursing homes, and the reason why is because that's where there is constant use of the drugs, right? Lots of antimicrobials being used, so the sensitive cells are eliminated and only the resistant ones remain, and those are the ones that get passed from patient to patient, especially if you're not being really careful about how you are moving from patient to patient and you're bringing infectious organisms with you. So when you hear the term multiple drug resistant, we're talking about a pathogen that's resistant to at least 3 of the antimicrobial agents that would have been used to treat them earlier. And cross resistance often happens because drugs are similar in structure. We have a little cartoon over here that says, you're right, it's wearing a red cape and blue tights, but really no, organisms don't do that, obviously. They are called superbugs, however, when they are multiple drug resistant. So how do we prevent this resistance from occurring? It's really not that easy, but you do have to do a few things. We're never going to stop it. It's always going to happen, but we can slow resistance from occurring so that we can develop new drugs and new treatments before we're out of all options, right? So how do we slow this down? How do we slow resistance from happening? First is we have to make sure that the patient has a high enough concentration of drug for a long enough time. So when patients stop taking their drug because they start to feel better, right? Oh, I'm better, I don't need this anymore. Well, what's happened is they've killed off the most sensitive cells, but the ones that are a bit resistant are still hanging out. And if they don't keep taking their drug to kill those slightly more resistant cells, then the slightly resistant ones are the ones that get passed on to the next patient, and it gets a little more difficult to treat those. Okay, so we need to make sure that the drug is in high enough concentration. We ought to be, I should say, using antimicrobial agents in combination, especially for more dangerous drugs. So you want to make sure that the drugs act synergistically and not antagonistically. So synergism is when you're using more than one drug and they're enhancing the effect of each other. So if you look at this little image below, you'll see the 2 antimicrobial discs, and you see the dotted line that is demonstrating where the zone of inhibition would be if we're using just that one drug. But when you're using them both in combination, you have a larger zone of inhibition. So that is what we want to do. We want to make sure that we are using drugs that act synergistically and not antagonistically, where they are going to interfere with each other. All right, our next little cartoon, you have a dying bacteria. It says, I'm so cold, and it's more resistant fellow bacteria next to it saying, no, hold on, Johnson. The human didn't take its full course of antibiotics. You're going to make it, and we shall rise, right? So that's the whole idea, that the organisms, if you don't kill them off, are going to continue and pass on. This is just a little bit of food for thought. It's an image that I took from Science News a few years ago, and it's showing that where we're using antibiotics here in the United States. And we use a lot, quite a lot. It says there's 51 tons consumed every day in the US. Okay, 51 tons. That is a lot. Where is it going? All right, so a small amount really is going to crops. So they're using some antimicrobials to treat various crops as they're maybe possibly being infected. Pets, so our dogs and our cats, right? The veterinary use of antimicrobials is the second smallest, I would guess. And then here, the aquaculture, those fish farms, you have to use antimicrobials because the fish get infected, and there's actually quite a lot of antibiotic use there, but it's nowhere near what we use to treat humans. So human use is quite high. But look at this dark blue bar at the bottom. Isn't that crazy? There's an awful lot of antimicrobials used to grow livestock, and they use it almost prophylactically. They use it because if they treat the animals that they're growing for food with the antimicrobials, then they don't get sick and they grow faster. So that's why they use it. They use a lot more antimicrobials, which is a huge amount. And so there's been talk about possibly regulating that use and preventing them from using some of the antimicrobials that maybe ought to be reserved for humans, so that if humans get sick, that we have options to use, and they haven't been, we haven't created resistant strains during our, our farming practices. And if you look over time, in 1996, just tracking salmonella and their resistance to antimicrobials since 1996, this is just one organism causing an infection, right? You can see that the resistance, the percent of strains that were resistant to the antimicrobials is gone up quite a bit. So we want to keep track of this. Okay, and so then poor ASIP technique encourages hospital-acquired infections, and this is just illustrating how if you don't have good aseptic technique, you can transmit infectious organisms in the hospital from person to person. So actually here we have a microbiologist who is being very careless. He's dressed for BSL 4 type work, so this must be a very infectious agent. Yet he's got his gloves off, he's got his protective face gear off, and he's interacting with somebody through the window, and it caused an infection in that individual when he left the laboratory. It says, until Bob entered her life, Lisa never even knew the meaning of the phrase acute multiple drug resistant infection by a transgenic Staphylococcus aureus. So funny now to think back, somehow all these little things change when you date a careless microbiologist. So we all need to use good aseptic technique, folks, right? All right, have a good one.